Hello, my dear students. Myself, Dr. Pitambar Humane, founder Botany for You, and working as Professor and Head Department of Botany, Dharampeth MP Dev Memorial Science College, Nagpur, Maharashtra, India. I welcome you all in this online platform for the students of Life Sciences Botany for You. Friends, if you are new or not yet subscribed our YouTube channel, then kindly like, share and subscribe our YouTube channel Botany for You. If you want to associate with us through the Telegram group, then the link is given in the description so that you can join the Telegram group and can take the advantage of the literature assistance in the form of PDF as well as career opportunities in life sciences in India and abroad. Friends, in our earlier classes on genetics and molecular biology, we have already seen the structure of DNA, structure of RNAs. And in today's class, we are going to discuss about the genetic code. So let's see what is the meaning or actually the genetic code is. As we know, if you see the definition of the living organism, then we know that the organism is made up of cell, and the cell is structural and functional unit of life. Means when any organism do any function, then it is carried out by individual cell. It means that that cell must have the certain information that is responsible to carry out the different functions in the body. The organism may be the plant or maybe the animal or even the microorganisms, and hence it is considered the cell is structural and functional unit of life. Now this is the cell, that is the eukaryotic cell, in which in the nucleus there are the presence of chromosomes or the chromatin material, which is made up of nucleosome, that is the histone and non-histone proteins, and the DNA molecule, which are responsible for the formation of the chromosomes. It means that in the nucleus actually there is a presence of DNA, and in earlier classes we have seen the DNA is double stranded helical structure. If we see this DNA, which is made up of the sugar, phosphate and nitrogen bases, and the nitrogen bases which are present in the DNA, they are ATGC at Gandhi Chowk, that is the adenine, thymine, guanine and the cytosine. Means the four different nitrogen bases are responsible for the formation of the complete information of the organism for its whole life. It means that this is the only thing which is present in the organism in which there is a complete information which decide the fate of the organism, including the internal as well as the external characteristics. No doubt, the environment also play an important role in expression of the external characters. Now, if you see these nitrogen bases which are present in the DNA, that is ATGC, adenine, thymine, guanine and the cytosine. So there is a complementary nitrogen base pairing in between the adenine and the thymine by the 2 hydrogen bonds and guanine and the cytosine by the 3 hydrogen bonds. So such a type of the pairing which is present on the DNA is known as the complementary nitrogen base pairing. Means both the strands are joined with each other by means of the hydrogen bondings in between the complementary nitrogen bases. Means if on the one strand there is A, on the another strand there will be the T. If on the one strand there will be the G, on another strand there will be the C and vice versa. So it means that both the strands which are present in the DNA molecule, they are anti-parallel to each other. And hence now this sequence which is present on the DNA is responsible to form the particular language in the form of the code. How this information is used for the different characteristics, that is nothing but the transmission of this information into the mRNA and later on to the protein. If we see the different characteristics of the organism, that is because of the formation of the thousands of different type of the proteins, and these proteins are dependent upon the mRNA, and the mRNA are dependent upon the type of the nitrogen base sequence on the DNA molecule. It means that only these 4 nitrogen bases are responsible for the formation of the complete code of the organism. Means the relation in between the sequence of nitrogen bases on the DNA or the mRNA and the sequence of amino acids in the polypeptide chain or the protein, and that is nothing but the genetic code. As these 4 nitrogen bases which are present in the DNA are complementary to the mRNA, and instead of the thymine in mRNA or the RNA, there is a presence of the uracil. So this is the difference in between the nitrogen bases which are present in the DNA and the RNA. So let's see the different meanings of the codons and its type in the next slide. Now if you see the codon, now as we have seen, the DNA molecule is a double stranded structure, and both the strands are anti-parallel to each other. It means that here one of the strand is used for the formation of mRNA, and that template strand which is to be used for the formation of this mRNA will be complementary base pairing. Say for example, if on the DNA there will be the A, here there will be the U. If here there will be the G, here will be the C. If here the C, here will be the G. If here will be the T, here will be the A. So and so on. So such type of the nitrogen bases will be there on the mRNA. It means that this sequence of nitrogen bases which are present on the mRNA will be utilized for the formation of the protein, and the protein is nothing but the polypeptide chain, or we can say that it is the chain of the amino acid. And this sequence which is present on the mRNA will decide that which sequence of the amino acid will be there in the polypeptide chain or the protein. So actually the genes which are present on the DNA will decide the structure of mRNA or the sequence of nitrogen bases on the mRNA, and the sequence of nitrogen bases will decide the sequence of amino acid and naturally no doubt the protein. And the proteins are responsible for the expression of the characteristics of the organism. It means that which type of the sequence is on the DNA will decide which type of the character will be there in the organism. So if you see this sequence of the nitrogen bases on the mRNA, then we can see the definition of the codon. The sequence of 3 successive nitrogen bases present on the mRNA, that is known as the codon. Say for example, if this is the mRNA and these are the different nitrogen bases, so sequence of 3 successive nitrogen bases, so this will be the one codon. Again the next 3 nitrogen bases will be the another codon, and in this way such a type of the sequence of 3 successive nitrogen bases, that is known as the codon. So this is a structure of mRNA, and this will be 3 successive nitrogen bases present on the mRNA, and that will be the codon. During the protein synthesis, here these sequence of nitrogen bases on the mRNA are read by the tRNA, and the tRNA on which there is a presence of anticodon loop, which is to be used to read the information on the mRNA. So the sequence of these 3 nitrogen bases on the anticodon loop of the tRNA, which are complementary to the codon on the mRNA, known as the anticodon, because it is complementary to that of the codon. Means the codon is on the mRNA and the anticodon is on the tRNA. When the protein synthesis initiates, there is need of the particular sequence on the mRNA. Say for example, now this is the sequence of nitrogen bases on the mRNA, that is from the 5' to the 3', because earlier we have seen in the replication that the process is carried out in 5' to 3' direction, and the same direction is used for the transcription also and translation also, that both topic we will see in the next lectures. Now if we see this sequence, that is, this is the first codon, that is the AUG, then second one UGU, third one that is the GGC, then next one is the ACC, and so on. So this is the first codon, and the first codon or the codon which is responsible to initiate the protein synthesis, that is known as the initiation codon, and there is a single initiation codon, that is the AUG. So AUG is the only initiation codon, while there are the certain exceptions like in the prokaryote or the mitochondria. So GUG is also initiation codon in the mitochondria, but in all the other organisms, the AUG is the only initiation codon, and which is present at the 5' end, from which the protein synthesis initiates. Then the termination codon. Termination codon means the codon which is responsible for the termination of the protein synthesis or the translation, that is known as the termination codon. So say for example, here there is a presence of UAA, which is a termination codon, because there is no any tRNA which is complementary to these termination codons, and there are 3 termination codons, that is the UAG, UAA, and UGA. The first one, that is UAG, is known as the amber codon, then UAA is known as the ochre codon, and the UGA is the opal or the amber codon. So these 3 different codons, if they are present on the mRNA, then there will be the termination of the protein synthesis, and hence these codons are known as termination codon. So first one, that is the initiation codon, which is the AUG, then anticodon, that is which is present on the tRNA on the anticodon loop, then the protein synthesis by which initiates, that is known as the initiation codon, and the protein synthesis by the codon terminates, that is known as the termination codon. Then the properties of genetic code, this is important. Now the code is triplet. Now earlier we have seen there are 4 nitrogen bases, that is the ATGC on DNA, and AUGC on mRNA or RNA, and already I told you this is the difference in between the type of nitrogen bases which are present on the DNA and the RNA, that is the thymine is present in the DNA and the uracil is present in the mRNA. It means that we have only the 4 nitrogen bases, and these 4 nitrogen bases are responsible for the formation of all type of the codons. If you see the natural amino acids, there are 20 amino acid discovered from the different living organisms. It means that these 4 nitrogen bases must be responsible to specify these 20 amino acids. So there are the many experiments regarding the deciphering or the cracking of the genetic code. So first one, if we see that mathematically, if we calculate that if it is made up of the one nitrogen base, that is A will form the one codon, then U will form the another codon, G will form the third codon, and C will form the fourth codon. So it means that the one nitrogen base equal to the one codon. It means that here there will be formation of only 4 codons, and these 4 codons are not able to carry out or specify the 20 amino acids, because the code is not ambiguous. The code is not ambiguous, it is a non-ambiguous. Then if the code is or the codon is made up of 2 nitrogen bases, so the 4 to the power 2, it means that there will be formation of 16 codons. So if you see this 16 codons, again the 4 amino acids will left, because out of 20 amino acid only the 16 amino acid will be specified by these 16 codons. But if the code is triplet, that is it is made up of 3 nitrogen bases, then there will be formation of 64 codons. And if 64 codons will be there, now they are very sufficient to specify these 20 amino acids. So this is a mathematical system, but later on experimentally it is proved that there are the 3 nitrogen bases which are responsible for the formation of the one codon. It means that here the code is triplet and not the singlet or the doublet. So in such a system, this 20 amino acid will be specified by 64 codons. Earlier we have seen that out of 64, the 3 are termination codons. It means that the 61 codons are engaged in specifying the 20 amino acid. Means the one amino acid may be specified by 1, the one amino acid may be specified by 2 or 3 or 4 or the 6. So more than one more than one codon can be used to specify the same amino acid. So such a type of the property of the genetic code is known as the degeneracy of the genetic code, that we will see in the upcoming slide. This second property of the genetic code, that the code is non-overlapping. Now say for example, this is the sequence of nitrogen bases present on the mRNA. That is, this is a 5' end and this is a 3' end. So if such a type of the sequence is there, no doubt here there will be the one codon, second, third, 4, 5, 6, 7, 8 and 9. So this is in case of the non-overlapping, means the one nitrogen base we are reading once. But if the same sequence of the mRNA nucleotides or the nitrogen bases is used as a overlapping codon, then first of all there will be the formation of this is the one code. If we use then overlapping, that is the first one we have to left and then again use, then there will be second one. Again here there will be the third one, then fourth one, so and so on. So in this way the number of codons will be the more. But again it is experimentally proved that if there are the 6 nitrogen bases, it means that in a 6 nitrogen bases there will be formation of here the 4 codons, but experimentally it is proved that the 6 codons or the 6 nitrogen bases are only responsible for the formation of 2 codons. It means that here the one nitrogen base is read once only, that is the codon is non-overlapping. The third property, that is the code is commaless. It means that there is no any space. If you see for example the AUG, it means that it is a initiation codon, And if we see AUG, that is the A, U and G, that is the methionine. So it will specify the amino acid and that is the methionine. Then here there is no any gap of the nitrogen bases in the form of the comma or no any other thing in between the codon. They are continuous right from the 5' end to the 3' end. Then GUG is there. It means that here the G, U and G. So it means that it is a valid. So it means that here such a type of the sequence of codons are present on the mRNA, and these different sequences are responsible to specify the particular amino acid, and in between the codon there is no need or there is not the presence of any nitrogen base in the form of the space or the comma, and hence the code is called as the commaless. The fourth property, that is the code has polarity, means it is read in a particular direction. As we know, the replication or the duplication is carried out in 5' to 3' direction. That is the formation of DNA from the DNA. Again, this sequence of nitrogen bases on the DNA is used for the formation of mRNA, that is the transcription. So again, this is carried out from 5' to 3' direction. Again, this sequence of nitrogen bases on the mRNA is used for the synthesis of protein, and again this process is carried out from 5' to 3' direction. It means that the everything which is carried out in this process or the expression of the genes, it is carried out from 5' to 3' direction. Say for example, if there is a presence of mRNA having the sequence AUG, UGU, then GGC, ACC, then UGC, etc. So then there will be the AUG will responsible for the methionine, then UGU for the cysteine, then for the glycine. So in this way, here there will be the synthesis of the different amino acid or to specify the different amino acid if it is read in a 5' to 3' direction. If we see in a reverse order, then that is the GUA. So instead of the methionine, there will be the specification of the another amino acid, and that is the valine. But when the sequence of nitrogen bases on the mRNA is present in the form of AUG from 5' to 3' direction, there must be the synthesis of the methionine and not the valine. It means that only the sequence which is to be used to read the information on the mRNA, that is the 5' to 3' direction, and hence we can say that the poles are read properly, and hence the genetic code has considered the polarity. The fifth one, that is the code is degenerate, or it is also known as the degeneracy of genetic code. Earlier we have seen that there are 20 amino acids and the 64 codons. Now see the amino acid, that is the methionine and the tryptophan, which are specified by only one codon. So if one codon for each amino acid, so one for the methionine and one for the tryptophan, the total number of the codon will be used, that is the 2. Then second one, that is the phenylalanine, tyrosine, then histidine, glutamic acid, lysine, asparagine, then aspartic acid, glutamine and the cysteine. So these are the 9 amino acid which are specified by the 2 codons each. So 2 into 9, that becomes the 18 codons will be required to specify these 9 amino acids. Then the leucine, that is the single one which is specified by 3 codons. So the total will be used 3 codons. Then again the valine, proline, then threonine, then alanine and the glycine, that is the 5 amino acids will be specified by 4 codons each. So 4 into 5 equal to the 20 codons will be used to specify these 5 different amino acids. Then leucine, serine and the arginine, that is the 3 amino acids, again they are specified by 6 codons each. So total 6 into 3, that is the 18. It means that 2 plus 18 plus 3 plus 20 plus 18 becomes now the 61 codons. And earlier we have seen that the UAG, then UGA and UAA, these are the termination codons. So plus 3 termination codons equal to total 64 codons that we have seen earlier in the triplet codon. And 2 plus 9 plus 1 plus 5 plus 3 equal to 20 amino acids. So in this case we know that except the methionine and the tryptophan, the rest of all the amino acids are specified by more than 1 codons. So such a type of the phenomena in which the single amino acid is specified by more than 1 codon, that is known as the degeneracy of the genetic code. And why there is possibility of the degeneracy of the genetic code, that we will see in the next slide. Now this is known as the Wobble hypothesis, which was discovered by Crick in 1968. So earlier we have seen the sequence of nitrogen bases present on mRNA is read by the anticodon which is present on the anticodon loop of the tRNA, and hence this anticodon and this codon, they are complementary with each other. Earlier we know that the ATGC, that is the 2 hydrogen bonds and 3 hydrogen bonds, they are complementary nitrogen bases on the DNA. In the similar manner, AUGC in the mRNA are complementary nitrogen bases. Means if on the codon there will be the A, then on the anticodon there must be the U, or on the mRNA if there is a G, then on the anticodon there must be the C. So if we see the Wobble hypothesis, it says that the first 2 nitrogen bases are complementary. Say for example, if this is a sequence of mRNA, that is the A, C, U. So if ACU is present on the mRNA, the anticodon will be UGA. But if the same sequence is read by the another mRNA, that is the ACU, then the anticodon will be UG, and here instead of the A, here will be the another nitrogen base. So which type of the nitrogen bases will be there? So if the anticodon will be there, then what will be the codon? So third nitrogen base on the codon will be complementary to this one. So A if there, then U must be there, but even if the G will be there, then again U can be present. So such a type of the nitrogen base pairing on the third nitrogen base which is not complementary, that is known as the Wobble pairing, and such a type of the phenomena is known as the Wobble hypothesis. If you see this one, that is the C and the G, then A and the U, then G and the C. So these are complementary nitrogen bases. But if you see this second diagram in which the G and the C are complementary, A and the U are complementary, but G and the U are not complementary. Here there should be the A, but there is a presence of G, or if here is the G, here must be the C. So, but these G and the U, they are not a complementary, but the first two nitrogen bases are complementary, but third is not. In such a situation, the third nitrogen base pairs with Wobble pairing. So, such type of the unusual pairing is known as the Wobble pairing, and this is the reason behind the degeneracy of the genetic code, that why more than one codons are used to specify the same amino acid. The sixth one, that is the code is universal. Whenever we are talking about the universality of anything, it means that it must be the same everywhere. If you see the different organisms from the viruses to the highly developed organisms that may the plants or the animals. So, it means that in the viruses also there are the presence of DNA and RNA as the genetic material. From bacteria onwards, the genetic material is present in the form of DNA, and the same thing is in the higher organisms, in plants as well as the animals. So, as we have seen that AUG, which is the initiation codon and is responsible to specify the amino acid, and that is the methionine. So, this AUG as an initiation codon in the bacteria also, viruses also, in the plants also, and in the animals also. It means that here the meaning of the same code in the codon dictionary is the same in every organism in the universe, and hence it is known as the universal. So, you have seen that the A, U and G, it means that the methionine. Again, the U, A, that is the termination codon that we have seen earlier. It means that in every organism the meaning of termination codon will be the same, that we have seen earlier, that UAG, UGA and UAA. So, these are the termination codon not in the one organism but in all the organism, and hence here almost that is nearly equal, because there are the certain exception that I have told you. In the mitochondria, the GUG is a initiation codon. So, except such thing, everywhere the meaning of the complete codon dictionary that is given here is same, and hence it is known as the code is universal. Then the last property, that is the code is collinear. As we have seen earlier, the information is read from 5' to 3' direction. That may be DNA, that may be mRNA. So, if there will be the synthesis of protein with the help of sequence of nitrogen bases present on the mRNA, it means that if the sequence of nitrogen bases are used for the synthesis of the protein, then there must be the similarity or the relationship in between the sequence of nitrogen bases on the mRNA and the sequence of amino acid in the polypeptide chain or the protein. So, say for example, if this is a one codon which is used to specify, then this will be the one. If this is the second one, then this will be the second. This is third codon, so this will third amino acid. This is fourth codon, so this will be the fourth. And in this way, we can say that here the sequence of nitrogen bases or the sequence of codons on the mRNA will show the same relationship with the sequence of amino acid in the polypeptide chain or the protein. So according to the sequence of nitrogen bases on the mRNA, there will be the sequence of amino acid in the polypeptide chain or the protein. So such a type of the relationship of the sequence of nitrogen bases present on the mRNA and the sequence of amino acids in the mRNA, that is known as the code is collinear, because it is present in the one single line and it is same. So friends, thank you very much for listening this lecture completely. I am very much hopeful and confident that this lecture will be helpful to you in preparation of your examinations. Friends, if you like this lecture and if it is useful to you, then kindly like, share and subscribe our YouTube channel Botany for You. And also press bell icon for the alerts of every lecture to be uploaded on different life sciences titles. And don't forget to join the Telegram group with the link given in the description so that you can take the advantage of the literature assistance in the form of PDF as well as the career opportunities in life sciences in India and abroad. And if you want to contact with us, then kindly visit our website www.botanyforyou.com. If you will have any query, then you can post in the form of the comments. Thanks a lot once again.